right, sorry I'm a couple minutes late here. How you guys doing? Doing all right? Okay. Thank you all for signing up for different topics. I've got that pulled up and we'll talk about that for a moment. Let's see, today we're gonna go kind of continue the overview of contaminants, but this time we're gonna kind of dive into how we would go and sample and discover what's in the water. And I think this will be a little bit of a shorter lecture today. So I'll have a few minutes if there are any questions about the research topics. There was a couple of you that had overlap, so we'll spend a couple minutes to talk about that, I guess after the lecture part. But overall, it was a nice fit in terms of the two topics that had two people sign up for that one. They're very broad topics, so it'll be very easy to kind of separate and say, okay, yeah, you take this sector of it and you take a different angle at it, and that'll work. Okay, so then let's see, other notes here to start the class. So yeah, I kind of mixed and matched a little bit here. We kind of talked on the sources a bit last time, and so this is maybe not the best labeling, but we're about on track schedule-wise. Mardi Gras is on Tuesday, so no class Tuesday, and then starting on the 15th, we'll get into okay, what kind of treatment stuff can we actually do, right? And so we'll start with some sedimentation and move on from there. All right, and then I think your thesis statements are due, so that would be your thesis statement and a one-paragraph your intro paragraph, including that thesis statement for your paper. And then in class, so that'll be on the 22nd, in class I'll have you each stand up and essentially state your research statement or your thesis statement in your own words, not in writing, so you're not reading this, but you'll stand up and say, here's what I'm going to write about. Here's the case that I'm going to make. It'll be something that you ought to practice for a few minutes before coming here and just have a little bit of something rehearsed so you're just not trying to read something, okay? So that'll be the 22nd. That'll be our first sort of oral communications thing. And then what you can do for your classmates, what I'll probably have us all do is give a bit of feedback on things that you felt like different people did well. You would not have to single anybody out, but it can be, oh yeah, I noticed people were projecting to start their sentence and found that very helpful, or something like that. Or, oh, this person, a few of you said, well, hello. And that was kind of nice that you said hello, right? So, something like that would be something we can look at. Okay, so that's just a quick check-in on where we're at. All right, so then, there it is, okay. So today, like I said, we're gonna talk about actually going and taking samples. What do we use? What kind of instrumentation can we use in the field? Chances are pretty good you've probably seen some of this stuff in the environmental lab or just in labs in general. But there's a few specifics that we can go and take a look at that might be pretty useful. We'll take to Nicaragua. In fact, I think I have a colleague that's going to join us from France, actually, and he's bringing an ATP measurement thing that will tell us something about how many microbes are in the water based on the presence of ATP. So that's kind of a neat one that I haven't yet added to these, but we'll make note of that in the future. All right, so then we have a couple things, right? Microbial and chemical quality for our water. And that's kind of for the drinking side of things. If we're checking to see if a source is reasonable to drink, we want to know how's the chemistry? Do we have arsenic in here? Do we have this, that, or the other? What's the deal? And then we also want the microbial. So a lot of times the chemical, we can use chemistry, obviously, and do some sort of a reaction, and that usually happens pretty quick. So there's a lot of things that are quick, and often we use some sort of colorimetric, meaning we cause a reaction and it forms a color, and the degree to which we have that color, we can see how much is there. So we'll talk a little bit about that. So this is, we'll say usually rapid. And microbial, aside from the cool chemical side of things like the ATP measurement system that I'm looking forward to learning about, usually you have to culture the microbes to see how many microbes are there. Maybe you can do some sort of a filtration and then use a microscope, but it gets very difficult to start trying to pick out all the different little things and counting that, quantifying that. It's quite the challenge to do anything that's immediate. And that's also typically impossible for viruses, anything that you do like that. So if you know you are looking for a very particular type of virus, maybe you can use some sort of antigen test or something like that. But at some point, if you're going to do this in the field, you don't have access to PCR instruments to check DNA. Maybe you don't know what type of DNA to look for in the first place. The microbial, portable microbial systems are typically going to be some sort of incubation, culture-based type thing, which means that's going to take probably overnight. So 18 to 24 hours time spent from when you take the sample to when you get a result. And this is actually pretty typical of what we do in drinking water treatment plants as well. We have continuous monitoring every day. There's checks, probably more than one time a day. And it's, okay, what's the influent in terms of any coliforms, and what's our treated water? Are we cleaning it? Do we have any massive load that is coming in from a rain event or something? Do we need to change or adjust our system? Having a good understanding and what's going on with that is important. Okay, so then there's considerations for things more like on the wastewater side of stuff, or perhaps even just checking on the quality of a river or stream. That, can it sustain life? What's impairing it? That's often related to wastewater discharges, but could just be general information gathering. So we might check things like the biological oxygen demand, so BOD, total suspended solids, of course pH, fecal coliform, so that again, that comes to our microbial. Oil and grease are a couple other typical water quality issues. We're not going to talk really about how to measure those. I mean, they become pretty apparent, but these are also the typical, I guess, the conventional pollutants we regulate and we treat for with wastewater. Okay, so then there are toxics that would kind of fall into the chemical category, and anything else. Okay, so chemical characterization. Often we'll look for kind of a suite of analyses. This will include the easy stuff, something you could just drop a probe into the water and it's got a fancy detector and says, oh, hey, we have this amount of pH. Oh, the temperature is such and such. There's some things that are very easy for a probe to do. And so partly because it's so easy, but also partly because these things like pH and temperature tend to affect rate constants and equilibria, all of that. It's good to just have a good characterization of what's in our water, or what the status of the water is. Like, are we acidic or what? So we've got a few that we can do with some sort of a probe. So conductivity is one of them. This is not quite total dissolved solids, but it relates pretty closely. So total dissolved solids would be anything that remains after you evaporate the water. So if you had a bunch of salt in there, you're going to end up with some solids formed because of the salt, because the salt's not going to evaporate. And it's also meaning dissolved. So if you filter it, the stuff passes through the filter. So total suspended solids would be removed by that filter. Whatever's remaining, maybe it's small organic molecules, maybe that are not volatile, maybe it's salt stuff. So that's our TDS. Conductivity is essentially a measurement relating to ionic strength, because what you do is you check how resistant the water is to electricity passing through. Okay, so the more charged ions are in solution, the less resistant the water is to electricity. So salt water conducts electricity a lot better than fresh water, right? So we can use a tool that just simply measures that and checks, oh, how salty or how ionic is our solution, and we can get kind of a generalized idea of total dissolved solids from there. Okay, so then temperature is pretty obvious. Dissolved oxygen is another one. We can use simply a probe. That's pretty convenient, and the temperature plus a dissolved oxygen measurement can often tell us, hey, are we close to being at equilibrium with what we'd expect from just oxygen in the environment? There are a few other things that affect the amount of oxygen that can dissolve a bit. For example, chloride concentration does impact the amount. So if you have a lot of chloride, I think that slightly decreases the amount of oxygen that your water can take. It's not a big effect, but it is an effect. So if you capture, maybe make some assumption from total dissolved solids, all that, you could get some estimate, okay, we expect to have this amount as our saturation value. And look, we're 3 milligrams per liter below that, so clearly something's demanding oxygen, where we've been oxygen deprived. Okay, ORP, the oxidation reduction potential. This is kind of a measurement of, in a sense, how corrosive, how oxidizing, or how reducing the water is. And this has to do, again, with the constituents in there. It relates also with pH. We often will plot pH versus ORP and get some understanding of, okay, at what point will something dissolve or change oxidation state in the solution according to where we're at with these. Okay, so all of these ones here are probe based. So we could get all of those with some sort of chemical probe, and that's very convenient. So they're almost instant. Put a measuring thing in. We have, maybe several different probes loaded onto one device or a couple devices or whatever, and we get the readings and we could record them. And that all takes a few minutes, maybe 20 or 30 minutes if you got to write everything down, right? So Eben does this, once a month with another grad student for a project we're on. 8 samples, 8 different spots on a couple rivers, and it's very familiar with that procedure, right? It's something that you can do. It can be routine. Sometimes you'll have it on a ship, and they'll have just something just in the water, and you'll stop at certain stations, drop it in, check, get all your readings, move on, check another, and get a sequence like that. Okay, so then we have all of those, and then we have colorimetric analyses. And if we're interested in something like nitrate, nitrite, sulfate, phosphate, arsenic, cadmium, fluoride, lots of different things, chances are pretty good that there might be some colorimetric method to check that. And in a field test, we will often have a nice little powder packet with all of the reagents we need set for about 10 milliliters of solution. And so it's going to have whatever chemicals react with, let's say, nitrite, built in there. And sometimes it's not a little packet. Sometimes it has to be like a liquid vial, and it's sealed off in glass. You break the glass and pour it in. And there's kind of fancy ways to do this, but there's a lot of them. And what you'll ultimately have is a scenario where you can cause that chemical reaction to occur given some amount of nitrite or whatever present, right? So in this case, it looks like this is nitrate being measured, and you'll see that color change increases with increasing concentration. And then what you have is a portable spectrophotometer, and your eyes are doing that spectrophotometry right here. So it's just a fancy calibrated way to do so, right? You've got shine a light through your liquid, and depending on how strong that light comes through, we could do this way. And you have a detector. So if my hand is the detector over here, you can kind of see some light gets through. Depending on how far it's traveling through there, we can calibrate that, how much light is being absorbed. And this should be water, so it's actually the bottle that's absorbing most of that light, right? But if the solution itself has something in it absorbing light, that's our measurement for it. So it's just a fancy way to do that. And we have portable ones these days with LEDs. It's very convenient, very lightweight, don't need a fancy lamp. We just need a good detector, which is just like the camera on your phone, right? So we have all of these pieces these days to make a nice, small, convenient device to do that. Now, each of these types of systems you can do in a laboratory with reagents. You can take these little powder packets, and it's fairly convenient if you need to check a few. You don't even have to think about it. You rip them open and do that. But that's actually a lot more expensive than actually creating a reagents and say, okay, now I need to mix X amount of this, Y amount of this, and do all of that. And so if you need to do hundreds of samples, you would probably want to look at the method, okay, they're using ascorbic acid and this, that, and the other reagent. Mix them appropriately and have this all set up in a lab, and it becomes rather quick and you can do it all. But if you're looking to just do a few samples real quick, even in a lab, we do this sometimes. We'll say, oh, let me just get some nitrate packets. Let's just use those. We're only going to do, 30 samples this year or something, right? So those are examples of things that we can do, and a couple ways to do them. The convenient thing, the neat thing is we can do that in the field very often. Okay, so just a little closer look at these types of things. These multi-parameter probes, this is kind of what I was talking about for the probe devices, and sometimes you'll have multiple probes loaded up into one cartridge like this, all feeding into this meter. Pretty much all of them operate in some way on quantifying how much electricity is passing through. So temperature probes, for example, check how easy is it for electricity to pass a certain spot that you put into the water or that you put into air, and that metal changes its conductivity slightly with temperature. That's the premise then. So if it's slightly harder then to push electricity through when it's warmer or colder or whatever, I think warmer would be the case, then oh, our electrical signal changed. Our default or our calibration at room temperature was this. Now we're a little above that. So it's requiring a little more voltage to get there. So almost all of these work on that type of principle. How is our electrical signal changing based on our probe's interaction with the environment? Okay, so there's fancy ways to do that. pH probes have a neat little thing that lets some pH across this membrane type of system, and then that interacts and you get that situation. The oxygen probes are similar. Conductivity is another one. That's actually just directly measuring through the water. So all sorts of probes are operating on that type of principle, and you get the fancy side of it gets to be what's happening on the inside of that actual probe. How are we getting that change in electrical current? Okay, so as you use these in the lab or in the field, keep that in mind in the sense that your electrical connection is really the important part. So making sure you have batteries and they're not going dead on you is probably going to be important. Making sure that you are taking proper care of the probe itself. pH probes, for example, you might think to yourself, oh, I need to clean this off. I just put it in some dirty water. Let's make sure we clean that off. Well, if you actually clean a pH probe with pure water and then let it dry out, that's a problem. If you let it dry out at all, it's a problem. If you decide, oh, I don't want to let it dry out, I've got some DI water here, let's put it in that, it's nice and clean. I don't have my probe solution anymore, that'll be good. That's actually bad. It would be much better to literally spit in the little capsule and use that as a way to keep your pH probe safe because of the ionic strength differences. So our saliva is going to have some amount of ionic strength, more than DI water. And it turns out that pH probes will lose their ability to function if they have all the salt pulled out of them. So the osmosis of DI water would pull the ions out of the pH probe and ruin it. So you don't store pH probes in DI water. So if you ever do that, let's not do that anymore. So normally you have like a saline solution to store them, and so you'll have a probe buffer solution or something like that. So you'll see the cap looks a lot like that, and then you have maybe a little bottle that screws onto where the pH probe ends or something, and you'll see it gets crusty and salty over time, and that's because it's so salty, and you splash and it dries up, and then you're getting the solids, right? Lots of total dissolved solids. So that's the proper maintenance for a pH probe. Requires some dirtier water rather than cleaner water. Of course, you're probably going to want to rinse it off before if you need to sample something else and you don't want to contaminate that sample, but a quick rinse with DI and storage in saline is fine. Storage in DI water is going to strip it all out, and that's bad news. So it's not a problem to take a measurement in a DI system, right? That's not the issue. Okay, so just a few things to keep in mind there. Usually these are fairly robust. This past year I've been learning a little bit more about nitrous oxide N2O detectors, and there's a very specialized one that's available, but it kind of goes bad after about 6 months. So there's something about the probe interface it's not stable for a super long time. So different ones, there are some fancy ones out there, but they're not all, always going to last forever. Okay, but the general ones, the generic ones that we get for kind of base water quality stuff are pretty good. And a lot of times these will also have a bit extra functionality. Sometimes they'll have GPS built in, so you can kind of say, oh, okay, this sample was taken here, and it just tracks that. It's like, oh, great. I forgot to check my phone or whatever, and this is probably especially handy before we had GPS on all our phones. It's like, okay, here's this spot. We've got the data logging, all that. And some of them you could even set up to do longer term data logging. You'd probably have something different than the actual meter like this. You'd probably have some device that's going to have maybe a solar panel if you're setting something up in the field and checking over long term, like months or years. You might just have a data logger sitting there pulling in the data, maybe a satellite connection to send you the data. Maybe you just have to go out there once a month, grab the data. So that kind of field analysis is pretty common. Then the colorimetry or spectrophotometry, could call it. The details here, I kind of mentioned them, but there's a specific chemical principle called the Beer-Lambert law that we know we can take a look at, and light will follow this law as it passes through some medium, right? So if you have something transparent, this is exactly my water bottle case, right? You have something transparent and some distance it's traveling through that transparent material. If something is removing a bit of that light or changing a bit of that light in some way, we can measure that effect or measure that quantity of the stuff based on how much that light has changed. This is actually why we see different colors of sunlight this is almost why, kind of why. We see different colors of the sunlight as you have a sunset or a sunrise. What's happening is you have a long distance, long path length of the light interacting with atmosphere. Now, the atmosphere is not really absorbing, so usually we use the Beer-Lambert law for light absorption. So the chemicals are literally absorbing the light, the photon, and probably just accepting that as some energy. In the atmosphere, we have something slightly different happening. We have scattering, and blue light will scatter a little faster or a little more often than the reds and oranges. So when the sun's traveling through more atmosphere, you have this scattering, so it scatters more of the blue light, and you end up with more of the other. So if you were to go up to the space station and you're looking at a particular region that would be experiencing a setting sun, it probably wouldn't look like a sunset colors because the light you're seeing is not passing through that much atmosphere, right? You're seeing it from a different angle, right? So now if you saw it, maybe if you get a spot where the sun is passing through a lot of atmosphere before reaching you in the space station, maybe you'd see it that way. But if it's like you're kind of over Texas and Texas is experiencing a sunset, you'd probably just see blue because the blue is scattering. So it's kind of interesting to think about that. Okay, so the way it works is we have some amount of light transmitted. We could call that T. And we can define that transmittance as the initial intensity of light divided by, or excuse me, the current or final intensity of light divided by the initial intensity. So this little example over here, you can see a laser shining into this, this sample of pink liquid, and you see right here, it also seems to be fluorescing, but that just helps us visualize it, so we're going to go with it. We can see that by this point, we don't really have much light passing through, like maybe a little all the way down to here, and then the light's just basically gone, right? And along the way, you see that the light is more intense towards the top, and it's decreasing. It's a neat way we can visualize this and really see that the light is being attenuated the further it goes. So we would define the amount of light that's reaching the top here. This would be the initial intensity. And then after some distance, we'd have I. Right, so we can take that current intensity after some distance divided by the initial and get some value there. And almost always in the sense of our little laboratories, our default path length for these cells is the little cuvettes, right? The little cute, like cylindrical vials that have clear sides, and you stick them in the instrument, right? You've probably all done this in chemistry or lab. And you put the cuvette, and that cuvette is a 1 centimeter square almost every time. So we can say that 1 centimeter path length is a default. But of course, you can actually extend that, right? The atmosphere, for example. Whenever you think about visibility, you hear, oh, it's a foggy day, the visibility is very poor, or whatever. Or you go snorkeling and you're told that though the water is so clear here, it's, you can see this amount through it, right? What you're talking about is, well, how far is that light going? Now, fog and turbidity and all that, a lot of that comes back to light scattering. It's not quite what we're using for, again, the absorption, but it's sort of the same principle about how far is the light traveling, right? Now, if you have clear ocean water, there's very few particles, and then you check how far light itself travels without any scattering, without any turbidity, you'll realize that, or you'll learn that red light tends to travel, I think it's red is the furthest. It's the least absorbed by the water. Blues will come out pretty early. And so you can go at different depths. And I should find a video for you on this, or a demonstration. But you could check, if you take like a rainbow plaque or something just to look at certain depths, some of the colors will just start looking like they're black, or just looking maybe gray. And so at certain depths, you'll notice, oh, we no longer can see this color. And after you get about, because the sunlight is not making it through. If you brought a flashlight, you could see the color, right? that would be fine. But the sunlight is just only making it so far. And so at some point, 100 meters or so, you're kind of getting no more light. It's all been absorbed by the water and the things in the water. Okay, so then A is for absorption. So, the absorption of light by the sample. That's going to we'll relate it to the intensity of light, how much light is still making it that far. And we can say the light absorption is negative log of the transmittance. So we can rewrite that. So let's say negative log of the transmittance. So that's negative log of the final over the initial intensity. Okay, so that would be the way we describe how much light absorption has happened. We can also define the absorption in the following way. We have a path length, that's going to be our C. That's how thick our cuvette is, right? If light's coming through, this is the 1 centimeter, typically. You'll find that if you need to increase the power or the sensitivity of your analysis, maybe you can stretch that into 5 or even 10 centimeters, but you have to make sure you have a light powerful enough to send light through it. So you can manipulate that. I'm sorry, B is the path length. C should be concentration, because that makes sense, right? Okay, so B is the path length, C would be the concentration of whatever it is you're trying to analyze, and then this last one here, this is our molar absorptivity. It's a coefficient. How strongly does your chemical absorb at a particular point, right? So I was working with Maddie this past semester. We were taking a look at something, a chemical that absorbs light. Let's say if we have light absorption, or we could call this our molar absorptivity coefficient. As a function of wavelength, the thing we're interested in absorbed a lot in the UV range, something like that, and then just a little bit over here near, I think it was 600 nanometers. And the deal was, this was one of these spectrophotometric processes, right? So it was one of these types of things where we had some reaction, we turn it a nice green color, and then the deal was the stuff we were trying to measure, we'll leave in red here, but the stuff that reacts to form that red, or for that green in that case, also had a lot of UV absorption in the UV range, but it did not have any absorption over at the 600 range. So we could select that 600 and say, oh, now that we've caused that reaction, all of that green color that we're seeing is coming from that change in light absorption. So that's kind of on the technical basis how we're going to do that. And then we're measuring essentially the color that's coming from that little peak in light absorption, from the change in the chemical itself. Then we can have a direct comparison to say, oh, if we can measure the absorption, which we can with these devices, right? That's actually what's happening right here. Visually, we're measuring that. We know the distance. We could make some estimation. Oh, I think 20% of the light makes it this far, or something, right? The instrument can do that for us much more directly. If you've ever played with like the pH strips and stuff and the colors and all that, it's doing that for you. It's assigning approximate values and letting you match with your eyes. So your eyes are doing that quantification because you have a nice chart, so to speak. So you have that like several different colors and you match the pattern to the closest one. You're actually doing all of that with your own eyes, that quantification, so we don't need a computer. They've just already pre-calibrated it to say, oh, here's what this color should look like. And here, you have 3 different colors to give yourself a good pattern matching, right? So the pH strips are doing exactly that and just using your own capability with your eyes to distinguish, right? Okay, so that's the premise here. The more concentration, the more color, right? That's the simplest way to say it, but I wanted to kind of give you a little more depth into thinking about how it functionally works in case you need to do some of these metrics. This also means that, say we're doing it in the field, in a lab, hopefully this gives you some appreciation for some of the things you might need to consider. Let's say you have a system where you lost the cap for your little device. And you can no longer shield it from light from the outside sources. Well, it's probably good to find a way to cover it, to shield it, because the light that you're measuring is supposed to be just that, the lamp that in your device before and after. And if you're leaving it open to the sunshine and somebody walks and makes a shadow, your measurement's going to be way off, because the light amount of light reaching that sensor changed quite a bit, right? So we, just thinking about it in terms of practically what's important, there's a lot there. Okay, so a little bit more examples. I mentioned these little glass ampule things. They're pretty neat, and they usually have a way where a system in place so that you can break them in a way that hopefully you don't hurt yourself with broken glass. But the sealing them in glass does allow the manufacturers to take a liquid that could very well be sensitive to oxygen and set it up in this glass sealed vial that's obviously watertight but also gas tight, and seal it in there with CO2 or nitrogen or some argon, something inert, something that's not going to react with your solution. And then they can, they'll ship that in a little styrofoam thing. Okay, so as we think about that type of thing, one of the major, and this by the way is a good example of one of these colorimeter devices, right? You have just kind of a little meter. Inside it, it has a light and a detector, and you're going to put your sample right inside here and put that cap over it and take your measurement. I can potentially bring some of these to the lab, to class if you're interested, but I think you've probably used things like that. Okay. All right, so then as we're doing our field analysis, there's a few other things to consider. Can we do the measurement right away in the field? If so, fantastic. If not, we probably want to put our sample on ice. Generally, we want to stop the reaction, stop bacteria from growing or dying. If we take a sample in Nicaragua on a hot day, it's pretty warm, that's conducive to reactions and bacterial growth and bacterial decay. All of that happens faster in warm temperatures. So we just chill it out, cool it off if possible, and go from there, right? So that's pretty standard protocol if you're going to do field analysis. Is you collect your sample, you take what measurements you can, you store on ice until further notice, right? Until you can conduct your analyses. So if you write a paper on it or a report, you generally would put in that protocol. Samples were stored, were put on ice, and analyzed within X number of hours, kind of thing, right? That's pretty typical. Sometimes you'll need to seal headspace if you have something volatile, if you're checking anything related to dissolved gases. So if that's something you need to concern yourself with for the analyses you're trying to do, that's quite important. And one thing to notice here, anytime you do field work, in developing contexts or not, one of the most important things is coming prepared with what you need to do, what you need to do, right? To label your samples, to collect your samples, to have some way of disposing of the powder packets that have trace cadmium in it, right? Or whatever. So some of these little powder packets have heavy metals as part of the reagent. It's not ideal, but it's what works, so that's what's used. So rather than just dumping those in the local landfill, is there a better way we can dispose those, right? That kind of thing is, there's a lot to think about in terms of being prepared. The sample bottles, the coolers, the ice, where are we getting all that, all of these pieces, right? Another part would be you may need to prevent light from entering your system. If you're worried about photosynthesis, if you're doing anything with BOD, for example, or if you're worried simply about your samples, maybe a breakdown of pollutants in there, right? There's a lot of photochemistry that happens if you leave water out in the sunlight. That will cause some changes. Sometimes maybe you need to acidify or perhaps occasionally add base to prevent some reactions from occurring, or to stabilize, or to prevent precipitation. Certain things based on your sampling needs, maybe you have to do that as you collect the sample. So several things to just be thoughtful about in preparation for doing field sampling. Okay, so the next type of indicator or type of test we would talk about is fecal and total coliforms. So this is, again, a direct callback to our interest in making sure that we have a good understanding of potential for pathogens, right? We have that fecal oral route of transmission. So if any bacteria that tends to grow well in warm-blooded gut, that's the type of bacteria we're going to try to incubate. And so we're going to give it conditions that are pretty representative of a warm-blooded gut, right? Something around 37 Celsius. So that's human body temperature, right? 98.1 Fahrenheit, somewhere around there. Or possibly a little higher to try to get a bit of a distinction. But in general, we can take, if we do it at 37 Celsius, that's probably going to give us what we call total coliforms. That's a decent indication of what might be living there. What kind of bacteria are there that would probably live in a warm-blooded gut. One caveat is that there are some microbes that naturally exist that would be happy to grow on this type of media that could be just naturally propagating in a bayou. Non-pathogenic, just heterotrophic bacteria, warm, aqueous environment with lots of nutrients around. So there might be some overlap. And so we try to distinguish it a little further and try to say, okay, well, because not all of the total coliforms are fecal in origin, and some places where we sample, maybe there's just a lot of total coliforms that are not necessarily fecal in origin. To try to distinguish a little bit further, we can do a fecal coliform analysis. These are defined as facultatively anaerobic, so meaning they are part of their process. Part of what they do is an anaerobic process. Typically, they would be rod-shaped, gram-negative, non-sporulating, so things like E. coli, and they can grow up to 44 Celsius. I know that E. coli is very unhappy if they go anywhere above 44. We sometimes in my lab we'll do a virus plaque assay where we infect E. coli with some viruses, and you have to heat a media up a little bit, about to 44 or so, maybe up to 48. And if you add the bacteria too soon before it cools down a little bit, you're just going to kill them all, like real quick. So they're very unhappy at anything hotter than that. So it actually does a pretty, there are some bacteria out there that would probably survive that, but some are pretty unhappy, at least to grow on a media that was hot like that. So in this case, you can add a little bit of heat, prevent some of them from growing, segregate that way. You can also do some things with dyes. So you see this figure on the right. You have some in blue and some in pink. So if you put something in your media that specifically this general class of bacteria are happy in one or take up one particular type of thing, and their biochemistry takes a certain route, that's going to end up showing up with the color or not, right? So we can make some distinctions that way. And in general, this is the sort of the end product, what it might look like one way or the other if we're doing some sort of count, and we have to find, okay, how many are here? We take a certain volume of sample, put it on a plate, and count it. Now, sometimes that putting it on the plate and counting it doesn't work so well. Sometimes there's just way too much, or you might not find any, right? So what do we do about that? Well, the more common problem is too much, because if there's not enough, then I guess perhaps we filter more water and catch them all on a filter. But if we're doing something with high concentration, then sometimes we'll have to dilute. So if 1 milliliter of wastewater has so many bacteria that it's just going to cover that entire plate, you're going to have to dilute it, right? Maybe you're checking drinking water and you have to filter 100 milliliters, and then you count 20, that's fine, right? And then we need a filter-based approach, which I'll show you one in a minute. So typically what we'll do is some sort of serial dilution where we take our sample, we dilute it, by maybe a factor of 10. So 1 part sample, 9 parts clean solution, meaning 10 parts total. So 1 in 10, that's a 10-fold dilution, right? What was 100 bacteria is now 10 bacteria in our vial. Then we can do the same thing and repeat it, and it's a serial dilution. A lot like a serial killer, just not as bad, right? A little hard on the thumb sometimes if you're doing a lot of them. But serial meaning in sequence there. So the math works out quite simply, and once you're working with this type of situation, it becomes it's a very streamlined thing to do to say, oh, we diluted by a factor of 10 to the 4th, meaning we did that 10-fold dilution 4 times, okay? And that's a 4 log change. All of that ends up tying into place in your mind once you've been working with it. So, just take a look here if you get at all confused about what I'm talking about, because you're going to do this and notice that, oh yeah, we went from 1 to 10 to 1 to 100 to 1 to 1,000 and so on, right? That'll all add up and be pretty typical. Okay, so you could take, for example, and answer this for me here. So what is the concentration of bacteria in a sample of water with the following results? And I'm asking, so maybe just grab some paper and do this as a quick thought experiment for me. So asking you to answer in CFU per milliliter. That's colony forming units per milliliter. And here's what result happened, or here's what the experiment showed. We took an aliquot, which is a fancy word for some drops, right? A little bit, a portion. A portion of water is taken from a sample and then serially diluted, 10-fold each step, 6 times. Then half a milliliter of that resulting dilution was spread across a plate. After incubation, you count 137 colonies. Okay, so I want you to come up with an answer for that. So just check yourself, make sure you're comfortable with this. This is the type of thing you'd have to do if you're actually measuring some bacteria. Some answers. Almost.
2.74 e to the 8. I don't think that's right.
I think that is right. Okay, so let me walk you through how I would think about this and see. I've done a lot of this stuff, so it might be I might be shortcutting it. But let me just say, if we had 137 colony forming units that we found, right, on the plate, that was in half a milliliter. So I'm going to divide this by 0.5 milliliters. And so we can, before we calculate the dilution, we can say, we'll just multiply 137 by 2. So that was your 74, right? Did I do that right? Colony forming units per milliliter. And I'm going to erase this. But after that's 10 to the 6th dilution, right? So to account for the 10 to the minus 6 dilution, you can just divide by 10 to the minus 6, or you could just multiply by 10 to the 6, and that should restore, like, that should be all you need to do, right? So imagine for a moment you had just diluted by a factor of 10. That's a 10 to the minus 1 dilution. So if this was a dilution by a factor of 10, you know, just based on your own, like, sensibilities, right? You know, if I did one part of this into, like, if this is 100 milliliters into 1 total liter, and I counted 100 in here, then there should be 1,000 in the total volume, right? That's just intuition. That kind of makes sense. You don't need to think too hard about it. It's the same exact process. When you've done a 10 to the minus 6 dilution. So all you really have to do at this point is say 274 times 10 to the 6 colony forming units per milliliter. And of course, that would be the same thing as saying 2.74 times 10 to the 8th. Does that make sense? Okay, so the reason I had you do that is at the end of the day, it's simple, but it's helpful to recognize how this is operating so you could, if you did count some on a plate, then you could make meaning out of it. So another thing you could do, and I don't think I have an example for you on this, is think about for a moment what happens if you had 137 colonies, but you had it on a filter that you incubated after filtering 50 milliliters of water. And then the question might be the colony forming units per liter, right? Almost the same exact problem. It's just a slightly different direction. This time we concentrated it by taking, I guess you could say per liter or per milliliter, whatever, either way. You can see that you're having to deal with the dilution effect, right? Or the concentration effect, right? With a filter, you're catching anything that was in those 50 milliliters. In a dilution scenario, you're diluting a bunch so that you can get, ultimately the point is to get to the point where we have a number of bacteria on a plate that we are capable of counting. Because if there's too many, trust me, it's just going to be a mess. And when you try to keep in your mind above about 200 as you count, you're going to get lost. It's not going to work. And the colonies will be merging, and you're going to get, it's just going to be trouble somewhere above 200 to 300 colonies. So if you ever tried to do that, you don't want to trust if there's just like 1 or 2, because sometimes they're a little sticky to the pipettes and stuff. So you generally want a range where you can count, where you're pretty confident there's maybe at least like 10, and no more than like 200 or 300. Like that's your sweet spot. Okay, so that's kind of one of the things that you have to navigate to do culturing testing. Add to that the complication that you don't know what concentration you have when you're taking a sample, right? It could be very messy or it could be very clean. So let's imagine for a moment we go to Nicaragua and we take a sample and then we also take a sample that we treat with some disinfection. Where do we check? Well, we won't know the data till the next day. So we actually have to check several things. We have to check a pretty wide range to begin with, and then if we're interested in seeing what happened with the disinfection, we have to check a pretty wide range after disinfection. And we're going to be doing a lot of guesswork. And that's just taking the one sample with one treatment. You could end up with 20 samples to run because you just are not so sure. So there's a lot to think about in terms of where and how to check and what all goes into that. The other complicating factor is, well, you at least have the answer the next day, but the sample will not be the same, right? Even if you store it on ice, maybe that's pretty close to the same. And that, in that case, that would probably be fine, to refrigerate it. But even then, there's probably going to be a little shift, right? So, or if you go out and sample the next day in the same spot, chances are good it's not exactly the same. So you try to capture it all in the same moment to get good data the first time around. So there's a particular kit that we use for this. We've got 2 of these in my lab, and on my recommendation, they ended up getting one for they're starting to set up a lab down in Nicaragua. So they've got one of these down there and can use them. So there's a pretty handy couple of YouTube videos on how these are done and how you count them. So we'll pull up one of these in a moment. I think this is probably, okay. So we might take a look at this. Actually, I think what I'll probably do is I'll try to bring it in so you can kind of take a look, play with it here next time we meet a week from now. And then watch this so you can kind of see, oh, how's this arranging, how's this working together. So that's, I'll make a plan to bring that in. Okay, so that's kind of, those are the major types of stuff we would get into, how to measure things. There are more ways and more tools out there. I didn't really mention chlorine testing. It's going to be another colorimetric thing. There's a few neat ways to do that. Kind of like the pH strips, you can get the chlorine measurements to have like a dial that says, okay, this amount of pink from this particular method relates to about 1.8 milligrams per liter of free chlorine, stuff like that. So that's actually, last year when I went to Nicaragua, we were using one of those and it was working pretty well. Okay, I think that's about all I wanted to say for this. Any questions on that? Okay, so we'll play with these guys a bit next time. And I've got just one more thing for you here. Let's talk for a moment about the topics. So overall, I was pretty happy with the spread. So we're going to have kind of a group for each. I will formalize the plans a little bit more, but basically we'll have one group that kind of teaches the class a bit more specifically about what's going on with groundwater quality, what can we do to predict or to map, what kind of concerns are out there for a few different categories. We've got a few people that will review different ion removal type of technologies. And so this will be a kind of I think a nice presentation you guys can put together for what can we do about it if we have too many minerals or whatnot. And then last, we have a few septic tank alternative strategies, specifically aerated treatment units, which I've touched on and Eben's working on as a project, so kind of cheating there. So I'll make him work for it and get some good info. But also, how about engineered wetlands? And that can tie in pretty well with perhaps some pre-treatment like that, or no treatment, or perhaps gray water or something like that. So why don't I have, there's 4 of you I'd like to just touch base with real quick. So that would be Sydney and Michael for the agricultural runoff. We'll chat real quick, and then Abby and Lauren. So if you also chat real quick about engineered wetlands, we can make some little plan on how to distinguish those a bit. And I think that's all. So have a safe and fun Mardi Gras. We'll see you next week. And the four of you just stop by real quick.